Hi, this is Andrew Wisegarver, and I'm submitting my project in the lab research category. I'm from Eastlake, and my project title is Novel Exon 3 in HEY1 as a product of the mutated DUX4 gene. Now, DUX4 is a gene that causes the Fascioscalpulohumoral Dystrophy that affects patients' face, shoulders, and humeral muscles, and can cause complete loss of muscle function. Now, DUX4 is mutated in this expression of FSHD, and it also mutates and creates a novel form of the HEY1 gene, which is known to be a signal pathway of muscle cells. So my project is to study a specific novel exon, one of 7, specifically novel exon 3, that was predicted to appear in the novel form of HEY1 by the computational biologist in the Tapscott lab at Fred Hutchinson Cancer Research Center. Now here you see the building that I actually worked in at Fred Hutchinson, and the Tapscott lab is there, and I worked with Jennifer Whidden, who's a graduate student at the University of Washington, who's working in the Tapscott lab at this time. Now, Jennifer allowed me to be very autonomous, providing me a guiding hand with my work, and though allowing me to learn a lot from my experience and prepare a lot of samples and of the protocols myself. And unfortunately, there were some parts of the protocol, some parts of the experiment that I was not able to do myself because of lab protocols, according to minors, and solutions such as ethidium bromide, which are deemed carcinogens. Now, this is the picture of the gel after the PCR reaction that was used to determine if novel exon 3 did transcribe into the DNA sequence of HEY1 inside a DNA sequence that came from a cell that expressed DUX4 very strongly. Now you see here there's the top band of the DNA in that 54-1 lenti DUX4 cell that we ended up using for the rest of the experiment due to its obvious highest concentration of any of the other samples, not including the GFP cells, which were known to be positive controls. And then also there's the bottom band of the DNA. Now those were obviously separated, cut out of the gel, and then extracted from the gel into DNA, which that DNA was then copied and cloned into E. coli cells and plated. Here you see the top band from that gel plated as E. coli cells, and then here's the bottom band. And then those cells, we chose a few and extracted the DNA from those cells, sent and then put the DNA through another gel, and then after that gel sent the DNA for sequencing. Now, the sequence data is here, which came from the Fred Hutchinson Sequencing Center, and it shows where the forward and reverse primers for that sample show, and then it shows in between what the actual sequence of the novel form of HEY1 that expresses the novel exon 3 where what that actual sequence was. And then here you see the primers and the actual sequence. Now, this is a picture in negative of the gel that was run with the top and bottom band DNA separately. The top band being lanes 1 through 3, the bottom band being lanes 4 through 6, and the idea there was to see where it corresponded with the previous gel and was the most concentrated, which were in lanes 1 and 4, which those were the two samples that were sent for sequencing the sequencing center. Then those two pictures were only different exposures in positive exposures of the the gel that you saw previously. Now, the most important part is this UCSC genome browser that my data was input into. Now, you see up at the top here, just for general reference, is the DUX4 ChIP-seq data from a previous experiment that was conducted by the Tapscott lab. And then you see here my samples compared against over here the novel Exon 3, its predicted site that it was predicted by the Tapscott lab's computational biologist. And you can see that the novel Exon 3's location corresponds with the location of my samples DNA, and that is proof beyond doubt that novel Exon 3 does exist in the novel form of HEY1, and it is a product of the mutated DUX4. Now, this research that I conducted is only a small part of continuing research in FSHD studies, and it will allow further research into possible drug therapies and ways to prevent binding sites and block binding sites by the scientists at in the Tapscott lab. And I'd just like to thank everyone in the Tapscott lab, especially Jennifer Whidden, for allowing me to come and work in your lab in the Fred Hutchinson Research Center.